Hello everyone, today my presentation topic is DNA fingerprinting. As we all know, the fingerprint could be used as a symbol to uniquely identify everyone in the world, so could DNA be such a powerful tool? It could be applied to find a criminal. It could also be applied in maternity or paternity tests like this. Okay, so the formal definition of DNA fingerprint is that this is a laboratory technique to determine the probable identity of a person based on the nucleotide sequences of DNA that are unique to individuals. Before I dive deeper, I would like to ask a question. So, why does all this make sense? How do we know that each other's DNA is unique? Well, actually, almost 99.9% of a human genetic information is the same with each other. It means that these three people are actually very similar. However, considering the huge amount of BP we have in one genome, 0.1% different actually means around 3 million BPs are different. That's a lot. That's why we could leverage that difference to identify each other's through DNA. So I would like to mention two popular methods to conduct DNA fingerprinting. The first method is called restriction fragment length polymorphism, RLFP. It actually is very powerful, but has a lot of limitation. So here, the second method, which is SCR analysis, is more commonly used nowadays. So let me first go through the process of RLFP. So step one, we should first extract the DNA from a person, for example, from a suspect of a crime. And then we use something called restriction enzyme to cut the DNA sequence. Everyone's DNA is different, so our DNA sequence will be cut differently, as you can see. And then we can get fragments with varying lengths. Secondly, we put those DNA fragments with varying lengths into a gel matrix, and we're going to go through a process called gel electrophoresis. The fragments in different sizes will move in the electronic current in different speed. Leveraging that feature, we can separate the fragments of different sizes. And then we put the fragments separated from step two to a special membrane exposed to a radioactive single-stranded DNA probe that has a complementary sequence to the target DNA of interest. For example, the DNA found on the criminal scene. And this probe will seek out and join the cut fragments from my sample, the sample from the suspect. Any excess probe that hasn't bound to the DNA of the sample on the membrane is washed away. And lastly, the membrane with the remaining radioactive probe is put to a photographic plate. The radiation will light the plate, and then we can visualize the sequence of bands which correspond to the fragments of different sizes. And then we can compare the bands of the target DNA and sample DNA, and we can draw the conclusion that whether the suspect is indeed the criminal or not. This is a simplified illustration of the result we get from the RLFP analysis. So here, as we can see, the two samples DNA sequence are indeed cut differently. So here, the first person's DNA is cut into a piece with 5K base pairs, and the second person is cut into two pieces with one being 2K base pair and the other being 3K base pair. Since everyone's DNA is cut differently by the restriction enzyme, the bands will look different, and we can leverage that differences to identify each person. However, RLFP has following limitations. So firstly, it's subject to degradation of DNA sample. Consider in this way, if the DNA have already been cut into fragments before the restriction enzyme cut them, then what can we do with it? Secondly, it requires a large amount of sample DNA. And here's when STR analysis comes into play. STR could solve the above because we only care about short tandem repeat region. So as long as those regions is intact, then we are fine, and we don't need as large amount of sample DNA as we do in RFP. And next, let me have a quick introduction of the STR. So STR refers to short tandem repeat, sequence of 2 to 6 base pair, and repeat many times continuously in a specific region. So for example, here, two person might have different number of repeats at one loci. So here, this person has 3 repeats of TGC. This person have 5 repeats. So 13 locations are recognized in the United States for STR. Please note that it's actually likely that you and I have the exact number of repeats at one location. However, according to statistics, the likelihood that we are the same at every location is 1 in a trillion. So here, let's go through the process. So firstly, we're going to do PCR to replicate the segments. And then we're going to go through the process gel electrophoresis again to separate STR of different sizes. By sizes, I mean different number of repeats. And then we'll get different bands. So let's come to the analysis part. Let me use a simplified example where SDR analysis is applied to a paternity test. So here you can see that for this child, one copy of the STR region corresponds to one copy of the father in this location, but this is not enough because it could be a coincidence that these two persons just somehow have the same number of repeats at one location. But if we conduct this analysis for other location and we find that, oh, one of the copy of the child corresponds to the father again, and then this one, similarly, it corresponds to the father's again, then what we can say is so the father is indeed the child's real father. Yeah, at least statistically. So here you may wonder why for this location the child only have one band. That's because the child is homozygous, during this region, so that the two copies have the same number of SDR repeats. That's why when being visualized, we can see only one band. Although very powerful, HDR also has its own limitation. So, for example, traditional method only support testing for repeat at one location, which is not fast, but scientists have been developing various software tools to handle the multiple loci simultaneously, which boosts the efficiency. The second problem is that mutation in the region around the STR region could lead to failure to replicate STR correctly. So here, one interesting thing is that although people might have different number of repeats in the STR region, somehow we found that the nucleotide around the STR region, which is called the flanking region, are actually almost the same across everyone. Yeah, so you can see here that the blue region between the two people are actually the same. That's why we can use a primer to bind to the flanking region to find out the location of the STR region. However, if someone has a mutation in the flanking region, then the primers might not work as expected, so that we might wrongly identify the region for STR, which may lead to problems. And there are some potential solutions. For example, we can use multiple sets of primers so that at least some primers will bind to the region that don't contain mutation. And then we can also conduct more analysis of more locations. So even though one location has some problem, the others will be fine. Yeah. So those are the potential solutions to the limitation of STR. And then in summary, in this presentation, I illustrate how powerful DNA could be as an identifier to separate everyone. And I also introduce RLFP and STR. And then further work could be done to improve the STR analysis. We could target at efficiency to enable parallel analysis of multiple locations. We should also target at accuracy so people can develop more advanced techniques to deal with mutations in particular. So that's all for my presentation, and thank you for watching.